All right, once again, I can't thank you enough for your time. So we were looking at this slide here that tries to model how DNA with 7,700 base pairs gets transcribed, then the primary transcript gets the introns removed, the exons spliced back together. You get what's called a poly A tail stuck on the 3' end. You get a what's called a G cap or a guanine cap, kind of a GTP cap stuck on the 5' end. But yeah, your final RNA is much smaller than the original DNA it was transcribed from. Yeah, now this is the model of how we make albumin, which is a protein that shows up in eggs and including human eggs. And it's a very, very important protein. It really kind of regulates our blood pressure. And yeah, we'll just show up. We'll talk about it a lot throughout this course. Okay, I don't know if you've never heard of an RFLP, a ref flip, a restriction fragment length polymorphism. Really what that, if you kind of look at this is supposed to be a chromosome. Do chromosomes really look like that? No. They're not nearly that straight. They don't have perfect lines. They're not that symmetrical. But you can see this banding pattern with the stains that we use. But anyway, going back to what this is trying to show, outside of these, I guess the bands are really where the genes are, or where we got the right stains to stick at least. Outside of those genes are these non-coding regions, but they really vary from person to person. So the value there is if we can figure out the structure of those non-coding regions, it helps us identify an unknown person, whether it's someone who left blood at a crime scene or DNA at a crime scene, or if you're trying to figure out who the father is. Okay. All right, once again, I love this picture, so hopefully you've looked at this and this kind of makes sense to you. We can see the cell, we can see a nucleus, we can see chromosomes. You can even see, portions or pieces of a chromosome with our light microscope. But you're not going to see individual proteins, histones. You're not going to see the double helix. Remember, we pretty much stripped all this DNA off of the chromosomes from the kiwi. Kiwi have chromosomes. And it just was that big snotty mess of goo as we pulled it out of each and every one of those millions of cells that were in the kiwi. Okay. Yes, now the model for replication. If you still have trouble understanding leading and lagging strands, let me know. Essentially, if you remember that polymerase enzymes read at 3 to 5, and it's really working towards the bubble would be the leading strand. So if you look at this picture here, the blue is the new strand being built. And it starts, or it reads at the 3, so the new one is built 5 to 3. So here's the new blue one being built, and it's the leading strand works towards the fork, towards the replication bubble. That little replication fork or bubble was created by helicase, which breaks the hydrogen bonds essentially. Primase kind of helps give it a place to start. So really there could be primase showing up down here too. All right, now if you look to build the lagging strand, it works away from the fork, away from the bubble. And it's still red 3 to 5, built 5 to 3, but as you build this lagging strand 5 to 3, it works away from the bubble. As a result, we get these fragments, and those fragments have to be ligated or joined back together with ligase. Quite honestly, even the leading strand has to be kind of joined or ligated in spots as well.